How's it going, everyone? It's Chow here, and today we are going to discuss a little bit about enzymes, catalytics, and thermodynamics. So let's get started. Alright, so the first thing that we like to talk about in regards to thermodynamics is this concept of entropy. So entropy measures the amount of disorder within a system. Obviously, you have an increase of disorder, meaning a positive delta S, or a negative change in disorder, meaning less disorder, and that's going to be negative delta S. And in terms of entropy, a positive delta S is more favorable. You want a reaction to have an increase in entropy or an increase in disorder. The second thing that we like to talk about is with free energy, more specifically G, or Gibbs free energy. And it's looking at whether the energy is able to do a certain amount of work and make a certain reaction occur or not. So we have two things. The first is negative delta G, and that's when a reaction is favorable. Sometimes we call it spontaneous, although that's not always the best way to describe it. But it's favorable, and it's going to be an exergonic reaction, which means it releases energy. On the opposite hand, we have a positive delta G, which is a reaction that's unfavorable or sometimes non-spontaneous, and it's often called energonic, meaning there's kind of an increase in the energy input needed to make that reaction occur. So if we have a reaction like this, you can have a negative delta G, so delta G is negative, and this is a lot more favorable, so energy release. And then if you look at the Gibbs free energy for this particular reaction, it's actually a positive delta G reaction, as shown here, and this is going to require an energy input, and it's less favorable. Obviously, you can run a reaction like this backwards, and if it's spontaneous or negative delta G forward, if you go back, it's going to be a positive delta G, which is basically this right here. So looking at the reaction and looking at the delta G, you should be able to see whether it's something exergonic or whether it's something energonic. So there you go. But when you look at it from the view of metabolism, obviously you have to look at it from two basic points of view, and that is what exactly are you doing to those certain molecules? Are you building it up or are you breaking it down? And in terms of metabolism, there are kind of two main types of reactions that you can do. And the first is a catabolic reaction, meaning one that you break something down. And then you have anabolic reaction on the opposite hand, which is where you're building something up. So a catabolic reaction requires often a release of energy, so a negative delta G, whereas with an anabolic reaction, it often requires an energy input, or a positive delta G. And if you look at the specific reactions themselves, you can see that with catabolic reactions, it's often hydrolysis reactions within the body, and biochemically, anabolic reactions often require the removal of an H2O, or dehydration reaction. So if you think about, for instance, the breakdown of glucose, you need hydrolytic reactions, and if you're trying to build up certain chains, for instance, if you're building up amino acids to make proteins, you need dehydration reactions. So these are just correlatory events and aspects of these two types of reactions. So when you look at these reactions, you often talk about this very high energy molecule that's involved, and that's ATP, or adenosine triphosphate. And ATP is a high energy molecule because of a couple of reasons, but a major one is because of the negative charges close together. So the phosphates have negative charges, you push them together, it's not very favorable. So it favors removal or hydrolysis or destruction of that ATP molecule. Now, when you have that hydrolysis, so when you have that phosphate group actually being broken off through that hydrolytic reaction, you're going to have an energy release. So when you have an ATP and you react it with water through a hydrolysis reaction, you're going to make ADP, or an inorganic phosphate, in addition to free energy. And so this free energy can be used to drive other reactions. So if you have a reaction that's maybe at equilibrium, so delta G is around 0, or maybe even delta G is positive, you can combine it with ATP hydrolysis to make it happen. So there you go. And I should just also know that sometimes you can actually turn ATP directly into AMP and a pyrophosphate molecule, and that can actually have even more powerful free energy drives because the delta G is actually more negative in that case. I think with just this particular reaction, the delta G is around negative 30.5, but if you actually turn it into an AMP, I think it's like negative 45. So it can have very strong driving factors for other reactions. So there you go. Now, when you look at enzymes themselves, obviously you can look at them as nothing more than just catalysts for certain reactions. Remember that enzymes, catalysts, aren't actually changing the delta G. So the delta G is still about the same. You're actually just lowering the activation energy of that certain reaction. So here you have an uncatalyzed reaction, and here you have a catalyzed reaction. As you can see, the delta G stays the same, but the delta G double dagger, as they call it in organic chemistry, or the activation energy, is lowered. So it speeds up the reaction rate, it lowers the activation energy, which is the energy it takes to get to the transition state, but it does not actually change the delta G or the equilibrium of the reaction. And also, enzyme catalysts do not cause reactions that would not occur spontaneously. So it doesn't really cause that delta G, if it's a positive delta G, to suddenly change to a negative delta G. So it really is just a catalyst to lower the activation energy. Of course, you can combine it with ATP hydrolysis, for instance, to make a positive delta G reaction occur, even if it isn't really necessarily favorable. But ultimately, the enzyme is unchanged by the reaction, so you can react it again. So in terms of enzymes, just a basic crash course as to how they work. Generally, they're going to be orienting substrates, they're going to be inducing some kind of a strain on the substrate, and you're potentially adding, temporarily adding chemical groups. So you have a substrate over here, and then you have an enzyme with an active site, and then through an induced fit model, you make an enzyme-substrate complex, which can cause the reaction potentially to react faster, and then this might be a method to lower the activation energy. Oftentimes, enzymes are specific to their substrate, and you react through this kind of induced fit model. Sometimes it's talked about as like a lock and key analogy, but in reality, it's more of just an induced fit, so like putting your hand in a glove. And this is basically how enzymes work to basically lower the activation energy and interact to form that enzyme substrate complex. So when you look at the reaction rates of enzymes, it depends on kind of two major factors. The first one is substrate concentration. So when an enzyme becomes saturated with substrate, meaning if you throw in a lot of substrate but only have a certain amount of enzyme, there comes a saturation point where basically the rate of reaction plateaus. So increasing the concentration of substrate doesn't cause an increase in the rate of reaction because the enzymes are already saturated. On the opposite hand, however, if you have a situation where you have a certain increase in the amount of enzymes, however, you can actually have an increase in the reaction rate in the presence of excess substrate. So if you're looking at an enzyme concentration versus rate of reaction graph in the presence of excess substrate, then you can add in more enzymes and that can increase the overall reaction rate. Whereas in this situation, if you have a certain limited amount of enzyme, but you just keep adding and adding more substrate, it's not going to make the reaction go any faster. So in terms of enzymes, there are kind of ways you can regulate enzymes. And one of the first ways is through inhibition. And so there are different types of inhibition available. There's an irreversible type of interaction where it can cause inhibition, and these are often through covalent bonds. So if you form some kind of a covalent structure on the enzyme through some kind of interaction, you're most likely going to make it irreversible. There are, however, methods to make the enzyme inhibition occur, but still keep it reversible, and that is through non-covalent interactions. The first type is a competitive type of inhibition where you throw in some kind of an extra inhibitor, and that inhibitor competes with the substrate for the active site. So you have the enzyme over here, and the competitive inhibitor actually goes into the active site of the area where the substrate is supposed to normally go. On the opposite hand, you also have other types of reversible inhibition, and one of them is uncompetitive. So uncompetitive inhibition is this instance where you actually have an inhibitor that binds to the substrate enzyme complex. And then finally, you have non-competitive inhibition, where you have some kind of an inhibitor that binds but to the enzyme on another site that's not at the active site, to kind of change the shape of the active site. So what's really interesting is that when in terms of inhibition, you can also have feedback inhibition, where basically the products of the reaction can kind of come back to inhibit the enzyme from producing more of that product. And we can see that, for instance, in glycolysis, where producing more ATP, the ATP can actually act as a source of feedback inhibition, where high levels of ATP will basically prevent glycolysis from occurring more and more. So there you go. Now, in terms of enzyme regulation, you can also have allosteric regulation, where you basically have a molecule that binds and changes the shape of the enzyme, or potentially inactivates the enzyme. So if you look at this situation over here, we have sort of a repression type situation where a substrate coming in normally would make products as a result. But if you have some kind of a repressor that binds to a certain site, it can prevent the substrate from coming in and therefore repress the enzyme and prevent it from producing the products. Whereas if you have an activation situation, so something similar occurs, but you have an activator, so normally the substrate can't bind to the active site. But if you have an activator that binds, it can change the shape of that active site and therefore allow the enzyme to come in and make products. So you can activate it or repress it depending on the enzyme type. And obviously, when it comes to regulation, you can also have other types of regulation. So finally, just a brief mention of pH. So pH can obviously affect the folding of the enzyme. If you have certain enzymes that have certain side chains, for instance, like lysines or arginines, or maybe like histidines or aspartates, glutamate side chains, they can have variation in their pKa's. So depending on what pH the whole enzyme is sitting in, so if it's sitting on a low pH, it might be protonated. If it's sitting at a slightly higher pH, it might be deprotonated. And obviously that pH can affect the folding and reaction of the enzyme. And so as a result, depending on what pH of the solution you're putting that enzyme in, it can ultimately affect the reactivity of that particular enzyme. And some enzymes have different preferences, and so if you have an enzyme that really is only active at low pH, if you put it at a high pH, it might not have an effect. So that's just kind of a crash course, a basic crash course on enzyme regulation and catalytics and thermodynamics. So I hope you all found that useful, and I will see you in the next video. And as always, best of luck studying, everyone.